Hello everyone, I'm Lakshan Beligala, a master's student from Bowling Green State University. My master's thesis is focusing on assessing geochemical processes governing phosphate sequestration in legacy phosphorus fields. This indicates a satellite image of Western Lake Erie Basin, so you can see the wide spreading of algal blooming. While considering the major reasons behind this scenario, agricultural excess phosphorus runoff is playing a significant role. According to the US EPA, this acts as the primary contributor to the water quality issues in US. Considering this pre-runoff, that can be happen from different kind of fields. But in my research, I am going to target legacy phosphorus fields. So what are these legacy phosphorus fields? What is the purpose of studying these legacy phosphorus fields? When we apply fertilizer into the fields, the main portion is going to be absorbed by the plants, and another portion is going to be withdrawn from the fields via different kind of losses, including erosion and runoff. At the same time, another portion is going to be accumulated within the soil since past and still it's continuing. That phosphorus is called as legacy phosphorus. According to the Bruegel, the fields which are having one or more sources of bioavailable phosphorus more than 100 parts per million is called as legacy P fields. According to recent research, in an individual agricultural field, 83% of the P load is contributed by the legacy P and the rest of the load is contributed by the incidental fertilizer losses. And at the same time, when we consider about the Western Lake Erie Basin, 33 to 66% of the total load in the basin is contributed by the legacy P fields. It means these legacy P fields are becoming a kind of hotspot in related to the agricultural phosphorus runoff from the fields. My research question: what geochemical processes govern the sequestration of bioavailable phosphorus in legacy P fields in Ohio? Now I am reaching to my hypothesis. Considering these legacy PVs, there are several potential reasons to have higher peak concentrations within these fields. The number one is higher clay content. At the same time, that goes to have higher cation exchange capacity and it goes to make more bonds with phosphates. The number two is higher organic matter content. As everyone knows, these organic matter have different kind of functional groups, including carboxyl groups, and that can be those groups can be deprotonated and facilitate more binding sites to these phosphates. And the third one is higher content of oxyhydroxide minerals because these minerals are having their own specific abilities to bind phosphates to their surfaces. Accordingly, in my research, I am going to target two major minerals, manganese oxyhydroxides and ferrous oxyhydroxides. Under the manganese oxyhydroxides, I am going to target bernicides. Under the ferrous oxyhydroxides, I am going to target ferrihydrides and garthides. Now you may have a question that why I am specifically targeting these minerals. I think I have an answer for that question in the next slide. According to the recent research conducted by our previous lab member Matthew Franks, he already found these minerals, it means manganese oxyhydroxides and ferrous oxyhydroxides, are playing a significant role in sequestering phosphates in the conventional agricultural fields. Accordingly, I have two specific aims. Number one is to investigate the geochemical processes involved in the sequestration of phosphates in legacy fields. At the same time, to assess the oxygen isotopic ratios of phosphates in three specific redox sensitive pools in legacy fields. Moving forward, now I am reaching to my methodology section. Actually, I collected soil samples from three fields under three different soil textures. Those are sandy, loamy, and clay. Actually, I cannot show any exact image or a location of these fields due to the confidential matters, but you can see the STP values. For the sandy, it's 652. For the loamy, it's 272. For the clay, it's 491 parts per million. So these values are way higher than the agronomic STP range. So that's why we are calling these fields as legacy phosphorus fields. Okay. This table indicates the STP values annual dissolved reactive phosphorus loads, and the annual total phosphorus loads from each and every field. So when you look at the sandy field, it has the highest STP value. It means the highest bioavailable phosphorus content in the field. However, comparatively, it has the lowest overall P runoff from the field with comparison with loamy and clay fields. And also, when you look at the clay field, comparatively, it has a lower STP value. It means a lower bioavailable phosphorus content in the field. At the same time, it has the highest overall P runoff from these fields. I think this is a very interesting scenario, and I have sufficient data to explain this scenario more in my result section. Okay. This indicates my sampling design. In each field, I collected soil samples from three different locations called location 1, location 2, and location 3. In each location, I collected soil samples from three different depths 0 to 5, 5 to 15, and 15 to 30 centimeters. Altogether, I collected 27 soil samples. So these are some images I captured during my sampling process. So you can see the 30 centimeter deep soil profile, then the step 1, step 2, and step 3. Then the next step was to conduct the solid characterization. Under this characterization, I already measured basic soil tests, total nitrogen, total phosphorus, total carbon, and organic carbon by using these different tests and instruments. As the next step of my research, I am going to measure heavy metals and cations. After the solid characterization, the next step was to conduct the sequential extraction. Actually, at this stage, my samples were subjected to a highly aggressive chemical procedure to extract phosphates from water soluble pool and three different redox sensitive pools, including manganese oxides, short range order ferrous oxides, and long range order ferrous oxides. It means specially targeting bernicides, ferrihydrides, and garthides. The soil sample I obtained at the end of the sequential extraction procedure is called as the residual. And it includes the phosphates we didn't able to extract by using the sequential extraction. And those phosphates can be highly bound to the other highly recalcitrant minerals in the soil. From each soil sample, I obtained four different supernames under four different pH conditions. Then I transferred those supernames for the geochemical characterization. As the next step, I am planning to transfer the rest of the supername for the isotopic analysis. After the sequential extraction, the next step was to conduct the supernance characterization. Under that characterization, I already measured total organic carbon and phosphates in each and every supername. As the next step of my research, I am going to target, I'm going to measure heavy metals and cations in each and every supername. Going forward, now I am reaching to my preliminary results. First of all, I would like to say this is still an ongoing research and these are just preliminary data. I didn't do any statistical analysis for my data yet. Here we go. This indicates the cation exchange capacity. As we expected according to the soil texture, it's increasing from sandy to clay fields. And same applies to the bioavailable calcium and also to the total organic carbon. When you look at the bioavailable calcium, in favor of cation exchange capacity, it also increasing from sandy to clay field. And when you look at the total organic carbon content, it also increasing from sandy to clay field. At the same time as we expected, it's decreasing as a function of depth because the activities that cause to increase the total organic carbon content in the soil, including microbial decomposition like activities, are usually high in the top layers than the bottom layers. However, I think these two figures are indicating the most important parameters. It means bioavailable phosphorus and total phosphorus content. The most interesting scenario is these two parameters are not indicating that increasing trend as we obtain in cation exchange capacity and total organic carbon. So when you look at the bioavailability content, the highest peak content is having in the sandy field than the loamy and the clay field. And when you look at the lowest bioavailable P and the total P content, both are having in the loamy fields than the sandy and the clay field. It means these are not indicating that increase in trend. In that sense, we can say these two hypotheses, it means the higher clay content and higher organic matter content. These two hypotheses are not 100% supporting my results. It means there should have to be an influence of other parameters for this having this type of scenario. I think these two figures are providing a highly possible answer for that question. Actually, these two figures are indicating the phosphate P mass percentage in each and every pool in both fields as a function of depth. So you can clearly see both manganese oxyhydroxides and ferrous oxyhydroxides are significantly contributing to retain sequestering phosphates in the legacy P fields. And especially when we compare the contribution of these minerals, that is way higher in the sandy field than the clay field. So that can be the highly possible reason to have the highest STP value in the sandy field and compared with the lowest overall P runoff from the sandy field. At the same time, a lower STP value and the highest uh, overall P runoff from the clay field. The overall conclusion is these minerals are playing a significant role in retaining, in sequestering phosphates in legacy phosphorus fields. As the next step, I'm going to measure the oxygen isotopic ratios in each and every pool to understand the original sources of phosphorus and also the rate of turnover in between these pools. As a conclusion, I want to say these phosphorus sequestering mineral complexes, it appears to be very, very crucial, but still it's an under-researched component of legacy phosphorus fields. This approach aims to generate valuable insights that could inform and also to take improved management strategies for the legacy phosphorus fields. In the acknowledgement, first of all, I would like to thank the major projects I'm already involved in. That's Hybrid Project and the Pilot Watershed Project, and also the main grant suppliers. And I would like to thank co-PIs, my co-PIs, Dr. Kevin McClooney and Dr. Angelica Vasquez Otaga. And I would like to thank Dr. George Bolejan and Dr. Carlos De Soto from Bowling Green State University. At the same time, I would like to thank Dr. Jay Martin and his team from Ohio State University. Finally, there's a huge hats off to my peer BGSO research team. Thank you for listening. And you can see my email address and the contact number. You're more than welcome to reach me to further questions. Thank you so much.